Hi, in this video I'm going to be talking to you about magnification and how to go about calculating magnification. Now, as far as the syllabus goes, this is what you should be able to do. Calculate magnification and size of biological specimens using millimeters and micrometers. So before we go ahead, just something that you need to be aware of and maybe make a note of is that 1 millimeter is made up of 1,000 micrometers, which are generally written with a Greek symbol, something like this. Or plain micrometers is fine. So this is somewhat like a U, except it's got a little longer kind of tail in front, you might say. Now this is also referred to as the letter mu. Not very important, not very significant in our syllabus, but no harm being aware of it. And therefore we say 1 millimeter is equal to 1,000 micrometers. Earlier this term used to also be called microns. So if you come across microns, it means the same thing. Okay? Or 1 micrometer is about 1,000th of a millimeter. Okay, now if you know this, conversions back and forth should not be too complicated. That's basic maths, right? Just keep that at the back of your mind. However, avoid using centimeters, use millimeters itself. So any time you're solving any of these problems related to magnification, make sure you're working in millimeters. If you've got something in centimeters, convert it before you do anything else. Okay, so let's begin. Let's take this image, let's say. It's the image of a pine seed. Here's the actual seed and here's the wing. But let's take the whole thing as a structure itself. This says into 4 or multiplied by 4 at the bottom over here. Now, what this indicates is that this image has been magnified 4 times from the original image. That is what this number states. This indicates, in other words, the magnification, or as this points out, that the original object is 1/4 of this image. Yeah, you can picture something like this. I'm just illustrating this for you so you get the point, that the actual object is 1/4 of it, and this picture has been magnified 4 times. In other words, if the length of this image is 8 centimeters, which we immediately convert into millimeters, which is 80 millimeters, that much I'm sure you're aware of, that 1 centimeter is equal to 10 millimeters. And then you measure the size of the original object, which we will call the actual length, which is 20 millimeters. Magnification is basically calculated as the measured length, or which can also be called the length of the image upon the actual length, which means the original length of the object. I hope you've got that. Yeah. Now this is an easier way perhaps to remember, because magnification MA is equal to M upon A, that way if you want to write it. But whenever you're asked to calculate magnification or one of these lengths, make sure you mention this formula, because there's generally 2 to 3 marks for calculating magnification. You're expected to show some kind of working. So in this case, this is how the magnification would have been worked out, and that's how this number 4 goes there. Okay, except remember, whenever you're doing magnification, magnification does not have a unit. So if you were to write this down as millimeters, you would obviously lose marks for it. Because obviously when you put 80 mm upon 20 mm, the mm also cancels out and you get magnification as into 4. Now if you understood this, typically in an exam, the kind of question you could be given is, you could be given this image with this symbol written down here, and then you would be asked to calculate the size of the original seed. I hope you've got that. Pause for a while, think about it, and make sure you understand this. So the question would give you a picture of the seed, and you'll be told it has been magnified 4 times, and you'll be asked, the question will be, find the size of the original seed, or the actual seed. So what we've got is magnification, so 4. What we've also got is the length of the seed. Now, how will you measure the length of the seed? Will you measure it from here to here? Will you measure it from here to here? Therefore, what you need to do is indicate to the examiner where exactly you're taking this measurement from. So for example, I would draw a line here and a line here and then a line all the way here to indicate to the examiner that this is where I've taken the measurement. And let's say we call this the measured length or the size of the image. This is 80 millimeters. I would write this on the diagram and I would also write this as measured length 80 millimeters. Of course, your writing better be neater than this. And then you put down your formula. Magnification is equal to measured length upon the actual length. Simple math there. Actual length therefore will be measured length, which is 80, upon the magnification, which is 4, which gives us 20 millimeters. Don't forget to add this unit now because we're talking about the length of the seed. So the original length of the seed is 20 millimeters. That's how this question goes. I hope you're clear with this. Now, what I'm going to be showing you now is a few variations of this question. Okay, if you haven't got this, go back over the bit that I've just explained and then proceed. Okay, let's look at another question. This is another kind of question that's asked. You're given a diagram and you're asked to make a drawing of the fruit. Now, whenever you're asked to do that, please remember, number one, your diagram should be much bigger than the seed. If you've been given a huge amount of space, use that whole space to draw the seed. And also keep an eye to see whether you are asked to label the diagram or not. For example, here it just says draw the fruit. You don't need to really label it. Sometimes they specifically ask you to label it, in which case there are marks set out for the labeling as well. Okay, so again, remember, number one, make the diagram nice and large. Number two, check if labeling is required. And number three, do not shade the diagram and draw it with plain, simple lines, somewhat like this. It should appear somewhat like this. Plain, simple lines, as against feathery lines. You should not draw kind of something like this. I hope you're getting the point. For now, imagine this is my drawing. Of course, that's more of a scribble, but let's imagine that's the drawing of the fruit. This is what the question says. Measure the length of the fruit in figure 1 and draw a straight line next to your figure. Now, whether this is asked or not, you are supposed to draw that straight line. If you remember, I told you, mark this point, and you can show where you have measured, whatever this number. Length of fruit in the figure, in this case, is 46 mm. And the length of the fruit in the drawing is 104, because that is what we've done. You mark out this point and you mark out this point. Not enough only to draw the main line, also mark out from where to where you're taking the measurement. And you've got this bit being 104 mm. That's the length of the fruit in your drawing. So remember, we could call this the measured length or the image length, and this is your actual length. Actual as per the figure in this case. Calculate the magnification of your drawing. You know this formula, which means measured length using the phrasing that has been given above, this one. You're saying it's length of fruit in drawing upon length of fruit in the figure, which gives us 104 upon 46, which is 2.3. And that's your magnification. Again, remember, there are no units for it. Hope you've got that. If you did not, pause the video, look through that once again. Okay, taking another example. Here's a different one. This shows a micrograph of a human blood smear. What you should be looking out for is any information that has been given, like this. And here's the question. It says, measure the diameter of the blood cell labeled A. Now, fortunately, this is a circular one. So, however you measure it, you should get the same size. But if it was this white blood cell, some could measure it from here to here. Others could measure it from here to here, which is why it's very important to mark where you're taking the measurement from here to here, and I'll draw a line here. If it were this cell, as I said, but that's not what we've been asked to figure out. Take this cell, mark this, mark a line to show where you're taking the measurement. calculate it and put it down. That's 5 mm. Please be careful. If you measure it in centimeters, convert it carefully. The photomicrograph has been enlarged 800 times. Okay, in case you did not notice that, they are being kind enough to tell you this. Calculate the actual size of cell A. Pause the video here, try it on your own, and then proceed to see if you got it right. Okay, you may pause it now. Okay, if you're done, check this. You should have written something like this, the formula. So actual length is measured upon magnification. You could have skipped this step and gone directly to this. That's also fine. Yeah, not a math exam. And therefore you calculate that the actual length is 5, which is your measured length, upon the magnification, which is 800. And therefore you get the actual size being 0.0065 mm. I hope you got this right. Now, what if you had been asked to write the answer in micrometers? Pause it for a while, and can you do that quick conversion? How do you convert this into micrometers? You're aware that 1 mm is 1,000 micrometers. Therefore, obviously 0.00625 mm will be equal to 0.00625 into 1,000, which will give us, if your math teachers taught you the shortcut, the 3 zeros, 1, 2, 3, which means I get a 6.25 micrometers. The same answer in my, I hope you got that. Yep, let's move on to the last question. Let's look at this one last one. Okay, this one, definitely try it out. Figure 6.2 is a photomicrograph of part of the fungus penicillium. Now, this itself should tell you that this is obviously a question from some board paper. You've got to make a large drawing of the structure labeled X, and then you've got to calculate some magnification. Now, I'd like you to try this out yourself. So perhaps pause the video here and try this out. This is a slightly tricky one, a little more complex than the other ones. But if you figured this out, then magnifications are pretty simple. So pause the video here and try it out. Okay, let's say you're done. And I hope your diagram is much neater than this one. I've just scribbled it quickly. You've got to make the large drawing. You've done that. You don't need to label it. That's fine. Now it says calculate the magnification of your drawing as compared with the actual size of the specimen that was photographed. I hope you got this point. They're not asking for this magnification. This magnification is showing you how much this photo has been magnified from the original fungus. Let's say here was the original fungus. This was magnified 1,000 times, and that's what this is. And now what you've drawn is you've drawn a magnified version of this diagram. What you're asked to find out is how much your diagram has been magnified as compared with the actual specimen itself. I hope you've got that. Otherwise, pause it, read it. So far, we were just calculating magnifications on one level. Here, like, you've got to go back to the original object itself. Okay, let's go. It says indicate on your drawing where the measurement was taken. So let's assume we've done this and you've marked this and you've measured this, let's say, as 100 millimeter. I'm just taking numbers that are going to be easy for us to calculate for now. And you are also measuring this bit and you find out this is 50 millimeters. So now if you've understood this question, we will need to break down our calculation into two parts. Let's first measure the actual size of the object that was photographed. Obviously, we don't know how big it was, therefore you can't mark it here. This was just something that I drew to show you that the original was something different. So now you've got the measured here, we need to know the actual here. You've also got the magnification. So the first part of our calculation shows magnification is equal to measured upon actual, and therefore actual size will be measured upon magnification, which is 50 upon 1,000, which will give us 0.05 millimeters. You're working in millimeters right now. Are we okay there? So you know that the original size of this now is 0.05 millimeters. And we know the size of our drawing is 100 millimeters. Now that should be simple, right? You've got to now calculate this magnification, which means the second set of working will be magnification is equal to measured upon actual. This time, remember, measured is 100 and actual is of the original object, which is 0.05, which gives us 2000, or our magnification is into 2000. That is the magnification of our drawing as compared with the actual size of the specimen. I hope you've got that. Okay, another shortcut that some of you may have thought of using, if you're able to understand this, great, otherwise ignore it, is that you will calculate only this magnification. You've got your diagram. I'll use a different color to show this so you don't get confused. If you've understood this method, please go ahead with this. But another way you could have also done it perhaps is your diagram has been magnified 100 upon 50. From the photograph, your diagram has been magnified 2 times. You know that this photograph has been magnified 1,000 times and your diagram has been magnified twice from the photograph, which means, simple math, if you're getting my logic, the magnification of your drawing as compared to the actual specimen will be these two multiplied, and that's how you would land at 2000 as well. Like I said, if you've got that, great. Otherwise, go back to the original working that we did down here. Either ways, it works fine. I hope you all are clear with magnification with that. As long as you draw large diagrams neatly, label them if required, get your formula right, show your working, and remember the units, I think you should be sorted.